Hello, this is Jim Schnabel. We're talking about oxidative phosphorylation today in our 26th in the series of biochem talks derived from the 7th edition of Principles of Biochemistry by Albert Lehninger. Today we'll be discussing oxidative phosphorylation. This is the route by which we develop ATP from NADH and FADH. Most prominently, and this is a highly membrane localized process occurring in mitochondria. These are convergence of all oxidative steps, and this is where we get our 2.5 molecules of NAD or ATP from NADH and 1.5 from FADH. There are transmembrane differences in protein concentration which drive the respiratory process, and there are three components to the chemiosmotic theory. One is that electrons flow through a series of membrane-bound carriers to reduce oxygen. Second, free energy between the electron flow and proton transport across the membrane is conserved. And third, protons move through transmembrane channels and they provide free energy for ATP. This function is critical to mitochondrial function. So mitochondrial structure is as follows. There are two membranes. An outer membrane is permeable to small molecules and ions, and the inner membrane is impermeable, and you have to use specific transporters. Mitochondrial matrix contains pyruvate dehydrogenase complex and citric acid, and this is where beta oxidation and amino oxidation occur. The mitochondria are numerous and high aerobic metabolism sites such as brain, muscle, and eye. And there are nucleotide-linked dehydrogenases, and they reduce NAD and FADH to NADH and FADH. These are reduced. And two hydrogen atoms are removed. One is a free proton and the other one as a reduced acceptor, and this is where you get NADH, NADPH, and FADH. The electron transfer occurs as free electrons, hydrogen atoms, and hydrogen ions. And there are three more electron-carrying molecules which exist: ubiquinone, which is coenzyme Q, cytochromes, which contain prosthetic heme groups, and iron-sulfur proteins. So this is an example of a quinone. This is ubiquinone, which is fully oxidized here. This is a side chain. This is a repeat unit of 10 of these moieties here, so it's a very long molecule. And there is a proton which produces a free radical. This is a semiquinone radical called QH. Free radical QH, and you get reduction of this hydroxyl group over here, retention of the aromatic ring. You can see it changes its location at this point where the proton is attached over here. Then you get a full reduction. This picks up a proton, a free proton, and you get the orbital filled, and then you get ubiquinol, QH2, which is fully reduced. Here's the heme series here. There's iron protoporphyrin 9, which is in the B-type cytochrome. Again, this is the iron and coordination complex with 4 nitrogens. Side chains are over here. There's also heme C, which is seen in C-type chromosomes. They differ with the side chain using cysteine linkages here as opposed to what occurs here with the methylene groups. And then there's heme A and A-type cytochromes where you get changes of the side chain and this long fatty acid chain over here. So the protein, the coordination that you get has increased complexity in A, the B, and the C, where the cysteine linkages attach to further and further higher numbers of iron, which are used in electron transport here. So there's 1, 2, and 3 as you get further complex with this tetrahedron moiety with irons occupying the corners and sulfur occupying alternate corners of this enclosure. A specific order of electron membrane carriers exists, and that's determined by applying inhibitors that act at specific steps, and these electron carriers are components of complexes. So you have the intermembranous space here, the matrix side over this end, and the main membrane arm, and you get extrusion of proteins as you go through the complexes. Here's a quinone, fully reduced quinone, and that picks up that will pick up protons as it goes through this pathway here, and through the matrix arm, and you get the NADH going back to NAD, and the protons are going through the quinone intermediate. The vectoral direction, protons are moving in one direction. There are toxins such as cyanide and carbon monoxide, which inhibit cytochrome oxidase. There are 4 complexes at work. One is the NADH dehydrogenase, succinate dehydrogenase, ubiquinone, cytochrome C oxidoreductase, and cytochrome C, then cytochrome oxidase. These employ combinations of flavins, iron, sulfur, and hemes in the complexes, and they associate tightly. Respirosomes are tight aggregates of electron transport complexes. Ubiquinone, that I showed earlier, can accept other electrons, including NADH, succinate, and fatty acyl-CoA. Electron transfer or energy is conserved in a proton gradient, which we saw in the previous diagram, and this is a reaction. The NADH reduced form plus proton and water gets NADH and water, and the energy transfer is very spontaneous at 220 kilojoules per mole. The free energy encompasses both the chemical potential and the electrical potential energy. The free radicals occur within the respiratory chain. The mitochondria are under oxidative stress, and superoxide dismutase removes that particular stress. In plants, especially in darkness, glycine is converted to serine and NADH, and NAD is regenerated through electron transfer to ubiquinone and later to oxygen, which was shown in the previous diagram. So for ATP synthesis, we have a chemiosmotic model where oxidative and phosphorylation are coupled. Again, the intermembrane space and the matrix on the other side. NADH is oxidized here. You get fumarate, succinate transition here. You have a proton pump that works over here, generating 4 protons, 2 protons there, and you get the presence of the superoxide reactions also occurring in sequence, the protons being converting and reducing quinone to quinolone right here. And the membrane potential is positive here and negative on the interior, and this helps drive the process through with the presence of production of ATP at the end line, and these are the names of the proteins that occur. So there's an alkaline chemical potential. There's also a charge potential here, where it's inside the interior is alkaline and negative, and the protons rather drive ultimate phosphorylation of ADP with transfer of protons in the process. So proton charge differences are what drives the ATP production. The inhibitors can be directed against electron transfer, ATP synthesis, or individual components of respiration. ATP synthase has two structural domains, F0 and F1. ATP is formed and converted to ADP on multiple occasions. The proton gradient drives ATP release from enzyme. F0 and F1 complexes have multiple subunits. They undergo conformational changes. Again, that's from the previous diagram. The active sites on F1 rotate and take turns catalyzing ATP synthesis, and the protons drive the rotation of the subunit. The process of chemiosmotic coupling allows for fractional stoichiometric yield of ATP from FAD, which is 1.5, or NAD, which is 2.5. There are shuttle systems which exist to convey NADH in the mitochondria. One of these is a malate aspartate shuttle, and the other one is a glycerol 3-phosphate shuttle. Electrons are transferred from malate and glycerol 3-phosphate, passes respectively to NADH and FADH. And for the ATP yield from glucose oxidation is a form of regulation. So again, going back to this, glycolysis, you get 2 cytosolic NADHs, 2 ATPs, and you either get 3 or 5 final ATPs from glycolysis. If you have the malate aspartate shuttle used, then you get 5 ATPs. If glycerol 3-phosphate is used, the yield is 3 ATPs. And then pyruvate oxidation, you have 2 of these, 2 pyruvates per glucose. You get 2 NADHs from that. That yield is 2.5. Acetyl-CoA oxidation citric acid cycle gives 2 ATPs or 2 GTPs, 2 FADs, which is 3, 1.5 ratio, and 2.5 ratio for the NADHs. 6 of those times 2.5 is 15. So you get either 30 or 32 ATPs per glucose as yield from this oxidative phosphorylation, complete oxidative phosphorylation of glucose. The pathway is dependent primarily on ADP substrate availability, so the ratio drives this reaction. Hypoxia acts on pyruvate dehydrogenase complex 4 in the respiration. The mitochondrial role, newborn mammals generate heat from oxidation of brown fat, which has a different histologic appearance. Fat under the microscope is basically like chicken wire. Brown fat has more of a speckled dot-like characteristic, and the lipocytes are not completely octagonal. If you look at these, if you look at brown fat on the microscope, this is not ATP, which is generating the heat. The protons do not pass into the F0, F1 complex. The protons are returned to the matrix. Steroid hormone formation is produced via cytochrome P450, and there's apoptosis, which is programmed cell death in tissue, and this is signaled by increased cell membrane permeability, and the proteases are released and digest the cell in question. The mitochondrial genome is quite fascinating. It's a double-stranded circular DNA molecule. It is derived from endosymbiotic bacteria. The DNA mutation repair is less efficient in mitochondria, and in contradistinction to males, mitochondrial DNA is inherited from females, as the eggs contain more mitochondria than does sperm, and mutations are conserved in mitochondria. So if you have, for example, if you're trying to track a particular gene through a family tree, the family tree transmission will be conserved through the female with the mitochondrial DNA, whereas the genome would reflect the contribution of the male in all the other available instances. We're talking about photosynthesis, which is an analog to oxidative phosphorylation. in this case ATP, is light-driven. The anabolic pathways predominate, so you get the carbon dioxide and oxygen producing carbohydrate and oxygen, and this is the reduction of carbon dioxide via the Calvin cycle after Melvin Calvin, who discovered photosynthesis. The process does bear analogy to oxidative phosphorylation. The chloroplasts are analogous to mitochondria in that this is an organelle, which is the site of the light-dependent pathway and carbon assimilation. Plastids are larger groups of energy organelles, and chloroplasts are a type of this, and they're subject to binary fission. So you have absorption of a photon, which has a quantum of energy, and that creates an excited state, which returns to the ground state through fluorescence. The chlorophylls absorb light energy, and they resemble hemoglobin. So chlorophyll A is shown here. There's a phytol side chain right here. This is chlorophyll B for this moiety right here. This is chlorophyll A right there, phenol side chain. There's a saturated bond in the bacterial chlorophyll at this point, and this is in bacterial chlorophyll has a difference. Instead of an alkene linkage, you get this structure instead. Accessory pigments extend the light frequency to absorb energy via beta-carotene. Chlorophylls funnel light in the reaction centers to convert them into chemical energy. And as a core complex of chlorophylls that surround the reaction center. So when we look at this, we have so-called antenna chlorophylls and carotenoids and other accessory pigments and a reaction center here. And impinging light will cause a photochemical reaction and convert the photon energy into a separation of charge and initiate electron flow, which radiates. This is the chloroplast right here. These are the reaction centers that you can see, and the electron cascade goes into the reaction center here. These molecules absorb light energy, and they transfer it between molecules. So the light energy goes into the reaction center. There's excitation, which releases the electron without fluorescent energy, produces a product. Reaction centers, there are two types. One is a phenophyton quinone. Electrons pass the cytochrome complexes. Multiple subunits occur. They're free radical intermediates and solid matrix. The other reaction centers are in sulfur. Same model with more enzymes. The kinetics and thermodynamics are extremely favorably efficient with an overall energy yield of 30%, and the free energy that gives free energy is quite low at negative 180 kilojoules per mole. So in more complex plants, these two types of reaction centers are integrated. They suggest a symbiotic facultative origin with oxygenic photosynthesis. So this is potential right here, the light impact. You have, again, a superoxide forming at this point with the oxygen evolving complex here. There is a P680 molecule. Light impinges on this. You get excitation. In photosynthesis, 680 is excited, and then you get a decrease of this charge. The proton gradient drops in this direction. You have these molecules in between a cytochrome complex here. With plasticyanin, you get excitation for the system 1, impinging light. Again, excited complex. The iron sulfur complex in this model. So there's one here and one here. These are integrated functions here, and at the end of this transfer, you get a presence of a reduced NADP molecule. The cyclic electron flow increases ATP relative to NADPH. Plants accommodate to different light conditions. Some systems respond to different wavelengths. The photosynthetic systems are separated and respond differently. The fate of water, you need to have readily available. It's a preferred molecule, provide the electron source. The electrons enter the pathway singly. The ATP synthesis is derived from proton gradient flow located in the thiol-alkaloid membrane, and the pH gradient drives the electron transfer. So again, you have impinging light. You get these complexes, PS2, with protons. You get a reduction of the quinolone complex here, and oxygen again is derived from the breakage of water with the oxygen atoms. So it's a free radical process, and you get a proton gradient, which drives ATP synthase here. And this again is the stromal side and the lumen side, the P side and the N side, and the plasticyanin. Again, this is another mechanism, impinging light here, and you get NADP reduced here, and this goes into energy synthesis over here. So you have a mechanism here and a mechanism over there. So per molecule of oxygen, you get about 3 ATPs and 2 NADPHs. The NADPH converts more ATP separately, as we've seen under oxidative phosphorylation. The ATP synthase is analogous to that found in mitochondria. Again, these are related organelles. A variety of obligate anaerobes use alternatives to water as electron donors. So you can have lactate, you can have hydrogen sulfide, and some plant species such as Halobacterium and Cyanobacterium have differing mechanisms for ATP synthesis. The Calvin cycle references carbon assimilations, where CO2 is converted to trioses and then utilized for broader synthetic pathways. And that's what this looks like here. The energy production via glycolysis, starch or sugar synthesis occurs here. You get ATP converting glyceraldehyde 3-phosphate to ribulose 1,5-biphosphate. You get carbon dioxide fixation at this point, going to 2 molecules of 3-phosphoglycerate, consumption of ATP with reduction by NADPH, removal of phosphate, and then glyceraldehyde 3-phosphate, which can go into the structure of a carbohydrate for structural or glycolysis or any other function, including sugar synthesis. So the overview of this has 3 steps. There's CO2 incorporated with ribulose 1,5-biphosphate by enzyme Rubisco, which is an acronym for ribulose 1,5-biphosphate carboxylase oxygenase, and this forms 2 3-phosphoglycerates, a magnesium ion, and a carbamylated lysine side chain coordinate the CO2 reaction with ribulose 1,5-bisphosphate. In the second step, the 3-phosphoglycerate is converted to glyceraldehyde 3-phosphate. This is a reversal of the glycolysis reaction. NADPH is used instead of NADH. There's a combination of 3-phosphoglycerate kinase and glyceraldehyde 3-phosphate dehydrogenase to get glyceraldehyde 3-phosphate. And then you regenerate ribulose 1,5-bisphosphate from trioses, dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate. This requires 10 enzymes and 3 ATPs per molecule of product. So in total, you have each triose phosphate is formed from CO2. There are 3 turns of the Calvin cycle, and that requires 6 NADPHs and 9 ATPs. This does not occur in the dark. This is a light reaction, and a transcript mechanism moves the triose phosphates out of the chloroplast, moves phosphates in, and you get sucrose produced in the cytosol. So photosynthesis occurs here. You have 9 ATPs. The Calvin cycle generates dihydroxyacetone phosphate. ADPs are produced here. These cycles, so there's an energy cycle of ADPs and ATPs here. Photosynthesis is at this point. The Calvin cycle produces carbohydrate, and this carbohydrate is moved out through a phosphate, triose phosphate antiporter. Phosphate replaces dihydroxyacetone, and this goes on to produce sucrose. There are 4 enzymes in the Calvin cycle which are activated by light. They cause protein and magnesium transport, which activates enzymes, and cysteine residues are broken. Photorespiration is a deleterious consequence of rubisco's lack of carbon dioxide substrate specificity. So you can combine oxygen with ribulose 1,5-bisphosphate to form 2-phosphoglycerate, which is a useless product. And this is can be significantly wasteful and exacerbated at higher temperature when oxygen is preferentially dissolved in solution. So there is a temperature range over which this can work optimally, and absent that temperature range, it will not work at all. So you have ribulose 1,5-bisphosphate. There's an anenediol intermediate with a double bond formed right here at C2, C3. And then enzyme-based or enzyme-bound intermediate is present at this point. You get this peroxide link right here, which will break immediately to 2-phosphoglycerate and 3-phosphoglycerate right here. So there'll be a cleavage point right at this point, driven by this reaction here. Two-phosphoglycerate is converted to glycine, then serine, called glycolate pathway. This and rubisco are pathways which conserve oxygen and produce carbon dioxide. The tropical plants have pathways to reduce these inefficiencies. The C4 pathway is one that acts at high light intensity and high temperature. C4 pathway traps CO2 as oxaloacetate, converts it to aspartate. There are 5 ATPs required to assimilate 1 molecule of carbon dioxide. C3 plants use 3 ATPs, a lower number per carbon dioxide, and the break-even point is about 28 to 30 degrees centigrade for greater efficiency of C4. For hot weather plants like cactus and pineapples, the CO2 enters the plant at night when the stomata are open during the cooler weather. So that acts at that point. We will stop there. Thank you very much.